Okay, this presentation is on the paper Cell Competition is Driven by Autophagy by Rina Nagata, among others. Um, I'm Derek Neufeld. Here's my email address if you need to get in touch with me. This presentation was made through CMU. So this study shows that the mechanisms that drive cell competition is linked to autophagy. Cells with compromised protein synthesis in the epithelial tissue are eliminated as losers of cell competition. Nagata demonstrates that prospective loser cells emerging adjacent to wild-type winner cells elevate autophagy levels, which induces NFKB-dependent HID expression and leads to cell death via the cooperation between HID and JNK. When this process is compromised, it can cause damaged or dangerous cells to grow unchecked, allowing cancer to flourish. That's why understanding this process and how it functions is vital in the fight to understanding and stopping cancer. So a main point of this presentation is autophagy, and so a little background on that here on figure 1. So autophagy is how cells get rid of maybe that aren't working properly or are damaged or things like that, and this is how they recycle them in order to break them down to parts that they can use to build new organelles. So along with just autophagy, there are also quite a few different terms that will be talked about in this paper. It's quite technical, and so I will go through some of them. This is not all of them, but it should give a little bit of a background. So cell competition is a quality control process that selectively eliminates unfit cells to ensure that their growth is stopped. Their growth is stopped in order to make the cells that survive in the body optimized and running at their absolute best. Autophagy is, again, the process by which cells destroy and recycle faulty organelles or entire cells in order to use their parts and get rid of them. JNK is a signaling pathway that, when activated, contributes to cell death through autophagy. So this is one of the main pathways that is being looked at in this paper, and how it mixes with the other pathways in order to create cell death. NFKB is a family of transcription factors that, when activated, also works to regulate cellular processes such as cell survival under stress, which, if this becomes corrupted, or if it's turned off, it can then lead to cell death. Since it normally causes survival, it can also do the opposite. CCP8 is involved in cell death and is activated by extrinsic factors from outside of the cell. CCP9 is the same. It's involved in cell death, but is activated by intrinsic factors from inside of the cell. HEL25E gets mentioned quite a bit throughout this paper. And these are the mutant cells, the mutant loser cells that they talk about. And they are cells with mutations in the gene helicase at 25E that leads them to be the so-called losers of the cell competition. That gets mentioned quite a bit because that's the main cell that is looked into in this study, so that will be mentioned quite a bit. So they used quite a few methods. Some of them are summarized here. So one main thing that was done were genetic screens. So there were genetic screens done to identify genes influencing cell competition, and so they used dominant modifier screens on Drosophila eye discs with these HEL25E mutant cells in a mosaic background with wild type cells. So it was mixed between the mutant and the wild type cells. That was used to identify some factors that could influence cell competition. And then some VATPase components were added to the genetic information of some of these cells and some of the mutant cells, and cell death rates were observed to drop after these VATPase components were added. And then another autophagy and lysosomal activity was tracked and recorded. So autophagic activity was tracked using ATGA mCherry, which is a protein marker that they used to track, and then they were able to see what the activity levels were for autophagy in these spots. Lysosomal activity was tracked using LysoTracker. So this is again just another tracker that they were using in order to see the amount of lysosomes that were used. I should add, lysosomes are used, you can see here in autophagy, they contain enzymes and are used to break down the organelles that are being recycled. So both were found to have elevated activity. Their borders between wild type and HEL25E mutant cells. Lastly, there was protein synthesis that they looked into. So by using O-propergyl puramycin, OPP, assay, it was determined that protein synthesis was reduced in the mutant HEL25E cells compared to the adjacent wild type cells. So the results from these methods were that for the VATPase activity, having the VATPase gene dosage suppressed cell competition, indicating that it has an essential role in the elimination of HEL25E cells, because when they have the amount of VATPase that was in the cells, then yeah, the cell competition was suppressed and the elimination of the mutant cells dropped significantly. They also looked at the borders specifically between mutant and wild type cells, and so they found that autophagic and lysosomal activity was elevated, which we looked at with the mCherry and the LysoTrackers. It was elevated at these borders. And autophagy inhibition at the borders suppressed mutant death, JNK signaling, and cell death. So, and also blocking JNK suppressed mutant cell elimination as well. And so they used the methods from before, and then HID regulation. So HID expression was induced at cell boundaries via autophagy and NFKB signaling, but it was independent of JNK. So they found that while NFKB and autophagy were directly correlated to HID expression, it was independent of JNK, while JNK still is a factor that leads to cell death. It goes through a different pathway and has to, or it activates alongside the other factors later on. They also found that autophagy was quite important. It was quite vital to this process. So autophagy was consistently elevated in loser cells at the borders with wild types, and the suppression of autophagy suppressed cell elimination in all cases. So yeah, every case, when they decreased the amount of autophagy that was happening at these borders, then cell elimination was eliminated. So this figure here shows the wild type and wild type cells here. So this is just a mix of only wild type, but then here in B, the wild type was mixed with mutant cells, and so you can see the wild type are in red, and you can see in this it's all red, meaning that the wild type like completely took over the entire eye, which makes sense because when mutant cells are next to wild type cells, then the wild type cells induce cell death in the mutants with lower protein synthesis levels, and so then they take over and then is left with just wild type. But here, since they're both wild type, just staying different colors, it's just a mix. And then in C here, you can see that in this eye, it's just wild type, and again, it's all the same color, but then here it is another mutant, and so you can see this is also all the same color, because when there's no wild type cells present, then even though there is a lower protein synthesis rate, and so if there were wild type cells, these cells will be eliminated. If there are no wild types, then the mutant cells, they just function as normal, and so these flies were with full mutant eyes performed basically as normal, as if they had wild type, so it seemed to not affect them too much, but the mutant cells completely took over and acted just as regular cells. And here you can also see this was fluorescently tagged to also see the differences. So the wild type mixture here again, it's just interspersed, but then here when it's mixed with the mutant, then it's mostly black, corresponding to the wild type there. So this study, it dives into a common mechanism for cell competition that can lead to cell death. This is centered around autophagy in mutant cells with decreased rates of protein synthesis when adjacent to wild type cells. These mutant cells have elevated autophagy levels, which induces NFKB dependent HID expression, which alongside J and K can cause cell death. This research is vital, as when this pathway is compromised, it can allow for mutant cancer cells to grow unchecked and wreak havoc on the bodily systems. So yeah, this is very important for cancer research, because when this process is not working properly, or it's damaged, then cancer cells really can just do whatever they want and can grow with nothing really to stop them. So figuring out how this works and seeing all the parts can help us get closer to a cure or a way to stop cancer deaths. So this last figure here, it just shows kind of the whole process, just in one figure. So here, these are the wild type cells, and these are the mutant cells. So when the wild type and mutant cells are close together, then the lower protein synthesis level cell, the mutant, it has the NFKB dependent HID expression here that is caused by possibly proteins coming from the winner cell, and then with elevated J and K levels and autophagy, then this works to kill the cell. But you can see here, there's another loser cell just beside the other, and this one remains unaffected because it only gets killed when it is beside a wild type cell.